Hola, Casey Bio. Okay, so today's screencast is on dihybrid crosses, obviously. And this is in contrast to monohybrid crosses, which we did in the last screencast that I gave you, which was about a week ago now, in which we looked at one trait, or I should say one gene, multiple alleles, and the inheritance patterns that follow. In this screencast, we're going to examine what happens when we look at two genes, each with multiple alleles. How does that work for inheritance? So, in order to discuss this, I think the first thing that's important to do is go back to talk about independent assortment. Okay, so if we have one cell here with, let's say it has two chromosomes, or two pairs of homologous chromosomes, so we've got the big chromosome and then the small one, and then that one has a pair, yada yada. I think you all know, or at least you should by this point, that we get, let's say this is a sperm cell, so we get 4 gametes, and that we have a couple different possibilities. If the chromosomes line up this way on the metaphase plate, all the resulting gametes will look like this. And if these two were to be switched, let's do that in black. If we were to swap these two, then an equally likely arrangement would be, or I should say which this is, that is an equally likely arrangement. We'd see something like this. The big chromosome, blue chromosome, small blue chromosome over here, and small red, big red. So, all of these gametes are equally likely. Now, let's talk about a certain gene that's on either of the chromosomes. So, the gene that's located here and the gene that's located here. Let's say that this gene determines shape of peas, so round or wrinkled. Round is dominant over wrinkled, so we'll say RR and RR. So, for example, this R allele is found at this gene locus on this chromosome, whereas the little, if it's red colored, we'll assume that's wrinkled and it's found in the same place, so the same gene, just different alleles of the same gene. Okay, you guys got that. And this one is color. Peas can be green or yellow. So the color gene is located again at the same place on these two chromosomes, different alleles. So the dark blue represents the green and the red represents the yellow. Okay, so you got that. And looking at all of the gametes that are possibly produced, we can either have a sperm that contains the big R gene with the big green gene, and that's both of these, right? Or we can have the little R gene with the little Y gene, and that's both of these. Or we can have the big R gene paired with the little, oh, sorry, this shouldn't be Y. This should be G, little g. Sorry, that's confusing. Little g is yellow. Okay. Or we can have the little R gene paired with the big G. And if you need to take a moment to think about that, how that's all possible, then please do so, because this is an important piece. Okay, so here are all of our possible gametes, and from this individual that is heterozygous. So I guess I should say that here. This person here is heterozygous. At both alleles. So they're heterozygous, they're big R, or I guess we should say, this is kind of confusing because the genome has already been duplicated, but before the genome is duplicated, this person is big R, big G, little g. Okay, so if you don't understand that, take a second to work through it and maybe watch that again. It's kind of confusing, but I think you guys can get it. Okay, so let's move over here to our P example, and I'm going to change the color of my stylus. Okay, so we got some P's. We've got a draw. Sorry, changing my thickness. Okay, okay. I don't know if I love that. That's okay. So here's our round green pea, and then, does that look wrinkly? Kind of. Close enough. Okay, so let's assume we have two peas, and we cross them together. So we have a round green pea and a yellow wrinkly pea. And they have pea babies, and all of these pea babies, are green and round. So they look like this. Okay, so not surprising, because remember before, okay, I guess I should say before we even do that, this is a true breeding green round pea plant, meaning all its parents were green and round, yada yada, way back, generations upon generations. And so it must be big G, oops, or I guess I should say the R first, big R, so both of its alleles for the round are, it's homozygous round, and it's also homozygous green for color. And this guy, same thing, homozygous round and, or wrinkled, homozygous yellow little g's. Okay, so if we cross those two together, the only gametes that these parents can make are, or I guess I should say, no, undo. The only the only pairing of round and green that we can see here is big G with a big R. This guy doesn't have any little r's, so you can only make round green gametes. Remember, let's say this is the father. He only makes these gametes, and the mother only makes little r, little g gametes. So all their children will be little r, big G, little g. So all of these offspring here have this big R, big G, little g. So each of them have the same phenotype and genotype. Okay, but let's imagine we cross these two peas here. The question is, what type of offspring do we see in the next generation? And this is where things get more complicated and kind of brings us back to all these combinations. So we said here before, with independent assortment, now you can mix up the R's and G's. So in the parents, we just saw big R, big G, and little r, little g gametes, but as you know, a heterozygous guy can make all the different pairings of big and little R's and G's. So now, when we make our Punnett square, it's a lot bigger. And this is the tough part. So, your Punnett square should look something like this. And if going back to these gametes, we need to put all these possible combinations around the outside, because remember, these guys are all heterozygous. So this genotype not only applies to this guy, but also this one, and this one, and this one. So when we cross two together, they all have the possibility of making the same type of gametes. Okay, so that means RG is a possibility. R, big R, little g, little r, big G, and little r, little g. All equally likely, and then the same thing down the side. Big R, big G, big R, little g, little r, big G, and little r, little g. Okay, so now this is the really annoying part. We fill in the Punnett square. Okay, so here we go. Big R, big big G, big. I'm just going to pause this. You guys can fill in your own, and we'll come back together when we have done it. Okay, so I filled in my Punnett square now, and actually I'm just going to erase this here and do a better line. Okay, that's a little better. Okay, so here's my Punnett square, and these are all the possible genotypes that you see from this combination, this cross of heterozygous gametes at one or at two different gene loci, two different gene places. And this is kind of overwhelming, but let's look really carefully at these genotypes and then determine the phenotype that we'd see from here. So remember, round is dominant over wrinkled and green is dominant over yellow. So this dude in the first quadrant up here is big R, which means he's going to be round, and big G, which means he's going to be green. So he's going to look something like that. And then move over here, big R, round, big G, still green. He's going to look like that too. Big R, still round, big G, still green. Big R, big G, still round and green. Okay, so I want you to take a moment, pause the screencast here, and I'm going to pause it as well and fill in this chart, and then we'll come back together when you've finished determining all the phenotypes that are going to arise from the genotypes you see in each box, and we'll discuss what that means. Okay, so here is the filled in chart now. And as you can see, the predominant phenotype that we see is round and green. Not too surprising considering those two alleles are dominant over wrinkled and yellow. But we also see some new combinations here. So I want to specifically point out these new combinations. Now we see round P that's yellow. Remember, not in the parental at all. And we also see a green P that is wrinkled. Again, not in the parent region at all. And lastly, we see a yellow wrinkled P, which again we saw in the grandparents, or the P1. I guess I should label these again. This is P, this is F1, so F2s. Okay, so here are possible combinations for F2. We can have green and round. This is the most likely, we see this most often. We also have green and wrinkled, and that is a possibility. We also have yellow and round. We see that guy. And we have yellow wrinkled. Boom, bada boom, bada boom. And these occur, so now this is no longer a mystery, these occur in typical ratios. So we can count them up here in our chart. Ultimately, we get 9, 1, 2, 3, 4, 5, 6, 7, 8, 9, 9 of these round green guys. This is what we'd expect to see. We'd see 3 of these round yellow guys, and 3 of these wrinkled green guys. Oops. 3. And just one lonely round yellow guy. So the ultimate outcome of a dihybrid cross, you're going to see a 9 to 3 to 3 to 1 ratio in the F2 generation. Okay, so this is a lot for sure, but I think you guys can totally take it and digest it and understand it. We are going to be working on some practice problems in the next few days on dihybrid crosses, and also talking about, this is simple Mendelian genetics. I know it seems complicated, but we're still talking about relatively simple modes of inheritance, meaning dominant or recessive. And we're going to talk about how that's not always the case, that sometimes we see genes or alleles of genes that interact strangely, or multiple genes are involved in one final trait or characteristic. Okay, but that's it for today. I hope you could see the connection between independent assortment and how important it is for understanding the possible gametes that can be made, and that really kind of underpins the whole understanding of where we see these different combinations of final alleles. Okay, so that is it, and yeah.